Alrighty, my name is Tanner Aldridge from Dixie State University. I'm doing this presentation for biochemistry number 2. This is the determination of genetic blood type, Rh and ABO. So clinical importance of Rh blood typing. The rhesus factor, Rh antigens, are expressed as part of a protein complex in the red blood cell membrane. Rh proteins may be Rhd, which means that they carry the D antigen, or Rhce, which means that they carry the big C or little c antigen and the big E or little e antigen. Rh proteins play a role in maintaining the red blood cell membrane. People who do not have Rh proteins tend to have irregularly shaped red blood cells and tend to suffer from mild hemolytic anemia. Most importantly, Rh antigens can elicit a large detrimental immune response. So HTR, hemolytic transfusion reaction, and HDN, hemolytic disease of the newborn. So in the case of HDR, if the Rh antigens of the blood donor and the recipient are not matched prior to the blood transfusion, the host's immune system will attack the unknown antigens, leading to the rupture of the donor blood cells. And this can lead to kidney failure, anemia, various lung issues, and even death. And then for HDN, or hemolytic disease of the newborn, anti-D antigens cause the most severe form of HDN. So if the fetus has the anti-D, but the mother does not have the Rh factor, then the mother's immune system will attack the fetal blood cells. And this can be a major cause of fetal death. It occurs when the mother's antibodies cross that placental barrier and attack the fetal red blood cells, as I previously said. This predisposition is just caused by the genetic recombination of the mother and the father, and it's just something that kind of happens by chance. So composition and clinical importance of the ABO blood types. So A, B, and O blood types are indicated by the presence or the lack of an A or a B antigen on the surface of the red blood cell. It is believed that these histoblood group carbohydrates are related to susceptibility of infectious diseases from our ancestors. The 4 blood types express the following monosaccharides as part of their antigen. So type A has N-acetylgalactosamine, type B has galactose, type AB has both N-acetylgalactosamine and galactose, and then lastly, the O type does not express any additional carbohydrates. And mismatching ABO blood types between the recipient and the donor during blood transfusion can have dire consequences. If a recipient who has, say, blood group O is transfused with non-group O red blood cells, the naturally occurring anti-A and anti-B antigens in the recipient's serum binds to the antigens of the transfused red blood cells, which then leads to rapid hemolysis. So all of the blood cells get ruptured, and the death of those blood cells can lead to blood coagulation, kidney failure, and even death again. HDN it can arise from this complication as well. It is a common disease related to the mismatch of blood type between the mother and her fetus. HDN typically does not require treatment because it tends to be relatively mild, mainly because fetal red blood cells don't express adult levels of A and B antigens. So, Rh and ABO blood type composition within the United States. So, what are the odds of somebody getting the right blood from a donor? Well, to get an idea, let's look at the national percentages for each blood type. Here you can see that Rh positive is 85% of the population, and Rh negative is 15%. You can see that A blood type is 42%, B blood type is 10%, AB blood type is 4%, and O blood type is 44% of the population. And given the high percentage of O blood type in the United States, it may be surprising that O is the recessive genotype. But if an individual has both A and O genotypes, they would express an A phenotype. Just as if somebody had a B and O genotype, they would only express the B phenotype. So the determination of genotype via PCR and endonucleases. So the recombination of PCR and restriction endonucleases can be used to elute the genotype of an individual. PCR, or polymerase chain reaction, increases the amount of desired DNA in a sample. After increasing the amount of desired DNA in a sample, you can add restriction endonucleases to that, and those can be used to cleave DNA at specific sequences to isolate and select for certain genes. After these cleavages have been performed, you can run the sample through a gel electrophoresis, and that can determine the size of the fragments created by the cleavage of the DNA, which helps you to kind of elute to what the genotype of an individual is. The amplification and cleavage of the D gene elutes to your Rh genotype, whereas the cleavage of an Rh positive individual shows banding at 1200 base pairs and 600 base pairs. An Rh minus individual only shows banding at 1200 base pairs. And then if we move on to exon 6, which is on chromosome 9, it contains the palindromic sequences for A1 blood type, A2 blood type, B, O1, and O2 genes. So the cleavage of this exon with KPN1 endonuclease shows whether or not you have the O1 genotype there. The O1 genes, if they are present and you cleave exon 6 with KPN01, you will show an additional banding there. If O1 is not present and you cleave with that endonuclease, you don't see any additional banding. If you cleave with, say, BSTE, that BSTE2, that endonuclease indicates the presence of A1, A2, B, or O2 sequences. So, given that, the cleavage on exon 6 really tells us if we have O1 or not. If we move on to exon 7, we can see that A1, A2, B, O1, and O2 can all be shown on exon 7 if it is cleaved with 3 different restriction enzymes. And the cleavage with those enzymes gives a distinctive pattern to each genotype with the exception of A1 and O1. A1 and O1, they will show up with the same banding patterns, which is why it is important to combine the results for an exon 6 and an exon 7 gel so you can determine if your banding shows A1 or O1. And then via that combination, you can really determine the genotype of an ABO person. You can tell what they are. So elucidation of blood type genotype for safe transfusion. So using PCR, restriction endonucleation, and gel electrophoresis to determine blood type, it's an effective method to ensure for safe blood transfusions, which can ultimately save lives. So here, if we look at the RH agarose gel, you can see that there is a band at 1200 base pairs and at 600 base pairs. So in this individual, in this instance, having these two bands show that we have an RH positive individual because you have your band at 1200 and your band at 600. If we move over to the exon 6 gel, we can see that there's no additional banding below 700 due to the KPN1. So that shows us that there is no O1 gene present in our sample. BSTE2 shows a band at 600, which is to be believed because it just shows that we have the other 4 genotypes in our sequence. And if we move over to the exon 7 agarose gel, we can see that we have banding at 309 base pairs, 204 base pairs, 145 base pairs, 119 base pairs, and again at 96 base pairs. So we can look at that versus a table that shows what the different genotypes have for specific banding in those areas. So further analysis of the exon 7 fragments. So fragments produced show that only A1, A2, or O1 can be present. So if we look down due to our fragments, keeping in mind that we have 309, 204, 145, 119, and 96, we can see that A1 and A2 in these green boxes, they line up where we would expect to see our banding. And then if we move over to the B genotype, that box at 223, we shouldn't see a fragment there, nor at 137 because that was not in our sample. If we move over to the O1 genotype, O1 was discounted from the beginning due to that KPN1 endonuclease that showed that we did not have O1 in our sample. And we move over to the O2, the band that shows up at 137 shows that this cannot be within our sample because we didn't have a band at 137. So our blood genotype is going to be A1, A2, and it's going to be Rh positive. So our phenotype will just be an A positive individual. So in conclusion, the genotype for this individual is an A1, A2, Rh positive, and his phenotype is going to be A positive. This individual can receive blood from A positive, A negative, O positive, and O negative donors because there's going to be no issues with the serum antigens there. This individual can donate to only A positive and AB positive individuals because those individuals will already have the antibodies and the antigens that this donor is going to donate, and his cells will not be attacked by the recipient cells. And if we move on to the next slide, you'll just see my references. Oh, I thought there were 6. I guess there's only 5. Oh, one of them was the Stanford Medicine blood right here. Didn't have an appropriate reference for that because it wasn't an academic journal. That is all.